Hello and welcome to the Providence College Podcast. I'm your host, Liz Kay, and I'm joined by producer Chris Judge of the class of 2005. Here at the Providence College Podcast, we bring you interesting stories from the Fryer family. This week, we're chatting with Dr. Jack Costello of the Department of Biology, who is finishing up his 30th year at PC. Jack, whose specialty is marine biology, has spent decades studying how sea creatures move. He just returned from a research trip studying comb jellies in American Samoa, thanks to a grant from the National Science Foundation. Jack, thanks so much for joining us and welcome back to Friartown.
Oh, thanks, Liz.
Well, can you tell us about your trip? What were you studying in American Samoa?
My goal was to study gelatinous zooplankton. And the reason that I went to American Samoa was because it's really out in the middle of the ocean. It's just this little speck out there. It turns out it wasn't as easy to study those planktonic animals there as I planned, because it's a little difficult to get outside the coral reef there. And the reef fringes the entire island, and the surf is quite high. So for a while we had a problem doing that, and then it turned out that we weren't really seeing the animals I was looking for. So I spent a lot of time free diving and looking at reefs and fish. We work not only on gelatinous animals, we also work on propulsion by all kinds of aquatic animals, including fish. And so it was a good chance for me to step back and look at how animals move in a wide variety of animals. So it turned out to be wonderful, and it was one of those times where I could just let the natural world tell me what was going on instead of approaching it with a series of tests and hypotheses that I had in mind.
So you could kind of go back to a blank slate and...
Yeah, and I think that's an important thing to do, because too often I feel that I approach things with a frame of mind as if I know what's going on. And in some cases I do, but a lot of times there's a much bigger story than the vision that I have of that. And so it was a very good opportunity to step back from that. And then we came back and were back in Providence for a couple of weeks and went down to Bermuda, to the Bermuda Institute of Ocean Sciences, which is also a reef system, but is much easier to get outside the reefs. And the animals that we're looking for are really there. And so we got a good chance to start on working on those and looking at it. I really wanted to see about it as a site going forward, because I'm involved in a program where we do field work on these organisms for the next 3 years. So I wanted to find a good site for that.
And why do this work in the field? Could you bring them back?
Sure. The whole premise of this project is the work has to be done in the field. Here's why. You consider laboratory work and what we learn in the laboratory, and we have learned a lot in the laboratory, but really very few animals are amenable to laboratory work. Very few animals can be cultured. Of the vast array that are out there, very few can be worked with in the laboratory, and that's particularly true for gelatinous organisms. These are animals that live out in the middle of the ocean, where there are no walls and never have been, and they use water as their support system. They don't have skeletons. They have very, what we would call flimsy bodies, and they live throughout the world's oceans, and in many areas they apparently dominate those. But now let's take a step back. We know that they're there. It's difficult to sample them because if you put a net down, it dices them up like jelly. So we know that they're there from particularly remotely operated vehicles and underwater methods. But how do we get any measurements about what they're doing? You can't put them in jars and take them in the laboratory. they don't react well. You can't grow them, first of all, no controlled experiments like that. So what we have done is modify the optical methods and measurements that we make in the laboratory so that we can do it all underwater. In other words, we can't take them to our laboratory, so we have invented ways to take our laboratory underwater to them. And so we need what I was looking for was a good site to do that. And I think we found one.
Bermuda.
Yeah.
Not American Samoa.
Well, not American Samoa. And it's really the place we went. I liked it because it was so remote. It was really pretty much of an outpost. Not easy to work at. Whereas Bermuda is, we may have found another good site in Florida as well. So I think we're in good shape to do this work. It will be demanding to do it because it all has to be done underwater. Doing work underwater is logistically really difficult. It requires boat support and travel time and a number of things. But I think we're pretty good at that.
I wonder, could you tell us how you got inspired to study animal motion and propulsion in the first place, and particularly in gelatinous animals? So we're talking about gelatinous organisms. We're talking about jellyfish and comb jellies. It started out from a very ecological standpoint. And my real question was, what are these animals doing there? What allows them to survive? What at times makes them really numerous? Just essential questions in ecology. We'd like to know who's there and how often and why. And so with jellyfish, one of the things that I noticed was that their capture of prey depended upon swimming, in the sense that the way that they capture prey is by moving water so that the prey run into their tentacles and capture surfaces. That was pretty much the beginning of being interested in the interface between animals and their fluid world. And once starting down that path, realized really needed to find methods that would allow us to, when I say us, my colleagues and myself, including former students, other professionals, really, we needed a way to observe that. And that meant we had to find ways to measure water flow around animals. And that was the beginning of it. And since then, it's just sort of followed its own path. Well, there's some really interesting applications to your research. I mean, what we're learning, what you're learning in your lab about the way jellyfish move in the water, how have people been working on applying that knowledge?
See, we really started with jellyfish because I was interested in the ecological phenomena of jellyfish.
Of jellyfish blooms, right?
Yeah, of jellyfish blooms. Like, why do they show up, when they show up, and what are they doing out there? And so we started to look at those very basic questions. And what we found as we developed methods to look at how an animal interacts with fluid was that, in fact, jellyfish were a very good model because they are one of the simplest animal designs. We tend to call them very primitive, and in an evolutionary sense, they're one of the earliest animal groups that we know of. Their record for forms that look pretty similar to current day forms stretches back over 500 million years. So they've been around for a long time, they've been successful for a long time, yet they don't have what we consider complex body forms like a number of fish. They don't have lots of fins and a variety of other structures, and they really can't produce force the way that muscles in fish do. So they're just not as complicated. And so when their bodies are moving, they represent really a very stripped down version of animals and the basics of propulsion and motion. So they've turned out to be a very useful, instructive model for animal motion and answering questions about whether flexibility, for example, of tissues affects propulsive design. They've been very useful for understanding how an organism maneuvers. And remarkably, it turns out that many of the principles seem to be similar for a wide variety of animal groups. It's just with jellyfish, they're so easy to observe and work with, and they don't have the complex array of speed and agility that a number of fish do, so it makes it actually easier to work with.
That's really neat. And in terms of the idea of just, this is the very basic kind of...
Yes.
It's one of the early, when you think about even more primitive organisms, they're often kind of static, right? But these move.
Right.
And so it's kind of one of the earliest forms of animal that developed with movement.
Yeah. We think that they're probably the first muscle-powered swimmers. Ctenophores are propelled by cilia. So they do have muscles, and there are some muscular motions they perform. But medusae, or jellyfish, which are, even though they're gelatinous, they're a totally different group than ctenophores or comb jellies. The medusae rely upon muscular motion to move. And so in that way, they're we think probably the first group to develop that. And they did so with what we consider very basic muscular tissues. They don't have the complex muscle structure that we have. They don't have the complex neural structure that vertebrates have. So in order to swim, they have to be very efficient. and for that reason, they've been a really good model.
And the things that you're learning about these really efficient swimmers, so they don't take a lot of energy, but a jellyfish bloom can occur and kind of take over a bay or a harbor or a body of water, right?
Right. When your body is comprised mostly of water and with very few complex tissues, that's a big advantage for converting what you eat into more of your body tissue. They're mostly water and very dilute organic material. And so they don't take a lot of organic material to produce a lot of body tissue. That's one of the advantages of a gelatinous design. It has some disadvantages. They don't have the agility or capability for a lot of the complex motions and activities that what we call more advanced animals have. But their ability to convert what they eat into body tissue is rather remarkable.
And there have been a number of collaborators who've been interested in kind of adapting what you've learned to apply to autonomous vehicles, for example, right?
Yeah, it's surprising to me to see at this point. Other researchers are using some of those principles to do really very remarkable work. And so it gives us a chance to contribute to a growing body of knowledge that is, it's a really gratifying thing as a scientist.
Well, if the Navy, for example, wanted to develop some unmanned vehicles to go underwater, some of these principles might be.
Yeah, and we call that transition from basic research, where we're looking at essential principles that underlie organism function, that's the realm that we're usually working in. But it can be translated into other functional designs and engineered designs. So we do some of that work. We provide some of the information to do that, and I'm on a project where we are working with engineers and an engineering firm to build a vehicle using, and the premise is that the efficiency of motion of jellyfish is useful, particularly for a vehicle that just wants to stay in one relative position for a long time. So we call that station keeping. And if you want to have a mooring, for example, that stays in an area, but it's in deep water, so it's very hard to actually moor it or anchor it. You can do that with propellers and all, but the problem is that those require energy and they have to be serviced quite often. And so one of the goals is to extend the period of time that you can put it out there without servicing it and refueling. So efficiency, energy efficiency becomes an important criteria. It's not as if you want to go someplace fast. If you want to go someplace fast, a torpedo is really a great design, right? But it's not energetically very efficient. If you just want to station keep, maneuver, stay in a general area, then the designs that we're looking at in nature are very effective.
So if you wanted to monitor the weather near, weather is the wrong word, underwater, the temperature or some other conditions.
Buoys that might go, that you might want to vertically migrate, that is, move up and down in the water column, sample in the water column, but come up to the surface, signal, send some kind of signal to satellite reception and stay out there and just keep doing that, much like modern day buoys do already, but just try and do it more energetically efficiently.
To reduce that kind of maintenance that you'd need.
Yeah, it's not so in this case it's no high secrecy covert operation. It's just an effort to build a more efficient system than what we have.
So Jack, you have done this work on all your research trips and you've traveled around the world to study these gelatinous creatures, but you also do your research on campus and at the marine biological laboratories in Woods Hole in the summers, right?
Yeah.
Can you tell us about what that's like to be a part of that scientific community there?
Great. It's a different environment than Providence College. Providence College we love because it's very contained and very focused, really. And the work that I and my students do here tends to be very focused on distinct processes that we can look at in the laboratory. When we go to the marine biological laboratory, the range of equipment is different, and the range of people we work with is different. That is, we have visitors from colleagues from California and Oregon and Florida and Brazil and Europe that visit us, and we work together on projects, and we have that laboratory. It has as part of it a number of optical approaches that we keep there that I share with my colleague Sean Colin. It's our joint lab. And so we bring oftentimes undergraduates. It depends on the project, the students, and the type of support that we have. But often we'll have students, postdoctoral scholars, other people, and it's more of an intellectual soup type of environment. There's just a lot going on, and we invent a lot of things there. New ideas, new methods, new ways to analyze data that we already have that's taken optically, but it's led to a variety of different kinds of results, and it's pretty unpredictable. So it's really fun. It's you never know what's going to happen.
Well, I was just about to ask you about the role of undergraduate researchers in your lab, and Sean is probably a great example because he's a PC alum himself, right?
He is. We worked together for quite a while, and Sean traveled with me at that point. I was working at Friday Harbor Laboratories out in Washington more. But then he went off and went to a master's program somewhere else, and then a PhD program somewhere else, and then went and taught. So he went and was very successful in all these other endeavors. But we stayed in contact, and then ultimately just started working together, and then just worked together as a pair for quite a while now, and maintain this lab together, and all of our projects are pretty much joint projects.
Did he come with you on your most recent research trips?
No, because this has been sabbatical. The poor guy has to work. Somebody's got to
teach those students.
No, this was, and he's had sabbatical and does things independently. So we share components, but we both have different institutions and different plans and timing. We just always try and share time down at the MBL working together, and we're in constant contact. Fortunately, Roger Williams University, is where Sean works, is very close. So we try and keep in contact. It's amazing how absorbing work is during the year, though. You think that just because we're close, that we'll spend a lot of time working that way. But really, once our semesters start, we're pretty much focused at our own institutions.
What else have some of the alumni of your lab gone on to do? Have you sent a lot off to grad school for marine biology?
Between biology and ecology, quite a few. There's oceanographers at NOAA in Washington, biostatisticians. The Providence Biology Department has a large number of medical students and medically oriented. So amazingly many of our students go there, but others also, Patty Schilling is a seal trainer at the New England Aquarium. So the diversity of what people do is quite impressive. Really broad array of things that people follow up and head into afterwards. Some go right into graduate school and in PhDs. Many people try out a variety of different jobs, and so we have probably great representation in biomed fields, but also ecology, oceanography. So it's pretty diverse, very impressive.
So if you think back at your own career, did you always think you were going to be a marine biologist? Was that your ambition growing up?
When I was a little kid, I wanted to be a veterinarian. And so I've always been interested in animals and nature. And then something happened to me, I'm not sure what, that led me more to field work. Part of it was as an undergraduate doing field work and working with some faculty that really made decisive differences in my career, and that was really influential. My undergraduate time was when I was most, unfocused. And I wasn't necessarily a good bet because my priorities were not always towards academics. And so I had actually a very spotty academic record, but some faculty members really took me under their wing, and that made all the difference. I I had great experience out in the field, and that turned my direction that way. By the time I got to graduate school, I was very directed. So it wasn't too hard for graduate mentors for me. To me, the fundamentally important place was undergraduate.
That's really interesting, because my understanding is that you didn't start off as a science major.
No, I didn't. I started off as an English literature major, and mainly because I liked reading and I liked writing. And so it was fun. And I took biology courses mainly because I was interested in biology. But I wasn't very interested in chemistry or calculus. I was just doing it for fun. And then at some point I realized I actually would prefer to do this. So I had a lot of catch up. That was in my junior year that I decided to switch majors, and I had to pile in all the courses. So the rest of my time was at college was fairly intense with courses, but I loved it. My senior year, I was taking 5 biology courses to fill in. And it was great. I loved that part of it. So that was pretty instructive to me about what I would enjoy spending my time doing.
There's probably some analogy there about being able to switch gears that we can make about being turn on a dime, which a jellyfish wouldn't be able to do.
Yeah, and I think partly it's one of the things I like about a liberal arts education, and why I believe in liberal arts education, the ability to think from a number of fields, and the mental agility to switch between them, I think, is quite important. At least I appreciated it. So, and I appreciated that I could do that because that's how much of a difference it's made.
Well, this seems like a perfect segue to ask you about, after so many decades of being a PC professor, you're also a PC parent as well.
Yeah.
So both your daughters attended PC.
They did. I think that they were the class of 2012, and but neither of them started at PC.
I didn't realize. Oh.
They both had attended public school in Providence, and even though they'd gone to all hockey games and sports here, we thought it would be best for them to go someplace else for college. And so they started at other places and moved around. But by sophomore year, both of them were back here.
What was it like to be on sort of the other end of the table as a parent enrolling your child here as opposed to being a faculty member? It's an interesting difference, because as a parent, you tend to be more protective. And as a faculty member, I had always scorned protective parents. So it was a conflict that way. But not much of one, because really they were off on their own, and they lived in the dorms, so they didn't live at home. And they weren't biology majors. So they were both over in the art department. And so we had to actually organize ourselves to intersect each other. It was very easy, and they just had a really good experience. So it was pretty easy to be a parent with kids at the institution. It was fun, actually. It was an interesting insight to hear about Civ and other things. Well, it's interesting because PC is a pretty small school, but the fact that you're both on a campus with less than about 4,000 students, and you still didn't have paths that intersected all the time. We would pass sometimes. And I remember one time my daughter Elise was with a friend, and Elise stopped to talk with me, and afterwards her friend said, well, who was that guy? And Elise explained, because her friend was taken aback that the familiarity that
that seemed to show in between us. But and her friend had no idea that her father was a faculty member. I mean, that was pretty new when she hadn't been here very long. Yeah.
Well, Jack, you've been able to travel all over the world and study animals in the ocean. I'm just curious, over these decades since you've started your research, how have the oceans changed, and what do you feel are the biggest threats to the ecosystems that you've been studying?
I don't experience a lot of the negative changes directly, because the places I go tend to be places that are more pristine or oceanic, or so it's not really something that I'm experiencing so much, except that we've done work that depends completely on climate change. We were looking at the seasonality of ctenophores in Narragansett Bay, and we're trying to explain why some years they show up very early. And as background, ctenophores, sometimes called sea walnuts, or they're predators, gelatinous predators. They're the non-stinging gelatinous organisms that are really numerous here, August through the beginning of October. If you go swimming down anywhere in the bay, you'll run into them, but they don't sting. Okay, they don't sting us. They're predators, and they eat crustaceans and fish eggs and essentially anything they can in the water column. And by doing that, they can have a major effect on the plankton community, which affects fish, and they can be a very dominant influence in the community structure. And our goal was to find out what is causing their variations from year to year. And one of the things that we found was that they overwinter in shallow waters. And so in years where spring started early and that were warm, they would warm up early and reproduce early, and then spread out into the bay and affect the planktonic community early. And that affected which copepods were present and whether there were copepods essentially. Copepods are small crustaceans that are an essential part of the community here and also serve as food for developing fish. So when the ctenophores show up early, they take out the copepods, and that influences what's left for the rest of the community as a food source. And what we found was, okay, warm years, it's pretty simple. It warms up their breeding grounds, and they are then available early throughout the bay. Cold years, on the other hand, they don't start until later in the year. And so there's a window of time where where the crustacean copepods can become numerous and develop and have what the type of community that was characteristic of Narragansett Bay 25 years ago, but is often not characteristic in present times because of these warming patterns that are particularly influential in shallow water breeding grounds. So actually things like climate change have almost inescapably become part of all of our research. Even if it's not the intent, it's such an ever-present component of systems that it's part of our work.
I'm curious what changes you've made personally in your own life, knowing that this is a factor.
Essentially, I try and live with as small an environmental impact as possible, and it's the changes that we've made When I say we, because it's my wife and my family all are part of it. A lot of the changes are really pretty easy. For example, we don't have a car anymore. I bike. We live in the city. We live on the east side, so it's not hard to do. And fortunately, in cities, there's ride sharing. Zip cars are completely adequate for us. So transportation is one thing. We try and live pretty simply. That includes food. We lead a vegetarian life, and so minimize our use of resources that way. Those aren't hard. Those haven't been really hard changes for us. It would be great if for transportation if we never went anywhere, but there's trade-offs that we make, and so those are some of what we've done. We just basically try and live a pretty direct and simple life within the framework that we have, and it doesn't feel like we're in any way restricted.
You may not be driving a car around town, but you're saving up the carbon for that trip across the world.
Right. I mean, that's where there's trade-offs. There's always ways to improve in what we're doing, and some of the decisions that we make are. We'd like it if there was a more energy efficient and less environmentally impactful way to do things, but in order to achieve other goals, we make the compromise that's available.
Well, you're one of among a good crew of PC faculty who bikes to campus, so I think you should feel proud that there are others making similar choices.
I'm glad to see it, and also these really brightly colored bikes from the bike stations that are around town are
the jump bikes. Yeah.
They look really interesting. So, and I've been very encouraged to see the rise in popularity of this.
Jack, thank you so much for being on the podcast. It's been great chatting with you about all you've learned in these years at PC. Subscribe to the Providence College podcast in all the usual places, including iTunes, SoundCloud, Stitcher, and Google Play, and our newest platform, Spotify. If you like what you hear, please review and share with others. Thanks for listening, and go Friars.